Hello, and welcome to Cognitum, a show dedicated to exploring the present and future of science and technology. I'm your host, Iosif Gershteyn.
Hello. Our guest today is Dr. Vadim Gladyshev. He is a member of the National Academy of Sciences, recipient of the NIH Pioneer, Transformative and Eureka Awards, Professor of Medicine at Harvard Medical School and Brigham and Women's Hospital, Director of the Redox Medicine Center, and the author of more than 450 peer-reviewed publications in scientific journals, as well as an inventor on multiple patents. Professor Gladyshev, welcome.
Yeah, I'm glad to be here.
So, Professor Gladyshev, tell me, what is aging?
Well, it's starting with a tough question. I would say that there is no consensus in the field on the nature of aging, and people view aging in very different ways. So, traditionally, aging is viewed as the increased chance of dying. So, for example, sometimes there is a saying like, we age if we are more likely to die tomorrow than today. However, mortality, people typically die from diseases. So some people say, well, wait a minute, mortality is just one feature and it's a consequence of diseases. Let's look at the incidence of diseases as the nature of aging. In a third group of people would say, no, diseases happen because of functional decline. So it's really the functional decline is the feature that defines aging. And other people say, well, functional decline is because there is accumulation of damage, like molecular damage in the cells. And so this is the defining feature of aging. Yet another group would say aging is a continuation of development. Organism, there's a developmental program to make a functional organism, and the trajectory kind of continues eventually and results in aging. So in the end, there is no consensus because people in the field, they basically view aging in different ways. If I, for example, ask you, what do you study? And you study aging. You say, I study aging. I also study aging, but maybe we study different things. So what I think aging is actually, it's an accumulation of negative changes of life. For example, molecular damage, but not only. So we live, we have functions, like various genes have functions, there are processes in organisms, but every function is imperfect. It produces negative consequences, byproducts of metabolism, mutations, epimutations, modifications of various proteins, altered levels of, hormones or number of cells in tissues. All of these negative consequences accumulate over time. And this is what aging is. So there is no like a main driver or the main process like in aging. It comes from the totality of the negative changes that happen as a result of life. That makes it really difficult to study and really difficult to interfere with. It's a very complex process. But I would stress that, again, so it's my opinion, and there's no consensus in the field, and the field is rapidly developing, and there's a lot of debates and discussions and many studies to try to address that question.
So given that aging is a very complex process, do you believe that there is a point at which aging begins?
Yes, definitely. So we know when, I guess, aging ends, when we die. But actually, it's a very good question. When does aging begin? I can tell you what I think again. But actually, let me start with this one. So 2 years ago, I chaired a Gordon conference, and we gave a questionnaire with like the basic questions to the experts in the field, like the questions you ask. What is aging? What causes aging? When does aging begin? Is aging a disease? Questions like that. And there was over 100 answers, and there wasn't a single consensus view on any of the topics. And the question on the beginning of aging was one of them. We basically gave them 9 options, and the people distributed more or less equally across all of these options. Some people think aging begins like at conception. Other people think it's at the stage of gastrulation, like early development. Other people, when organisms are born, like you're born, you begin to age. Other people think, no, it's the age of minimal mortality, which is 9 years in human. Other people think it's no, it's a puberty. So once we become reproductively active or are able to conceive, that's the beginning of aging. Other people think continuation of development, like around age of 20 or so. There are many options. So for me, the realization when aging begins actually happened when we applied biomarkers of aging, which were trained like late in life, and we asked a question whether they still tick early in life. And we could see that this kind of aging clock that quantifies the aging process actually already ticks during development. And we pinpointed this to the stage called gastrulation, which is we primarily study in mice, but in humans it would be equivalent to a third week of life. That's when I think aging begins. This period or this phase in life corresponds to the lowest biological age. So yeah, from that time point, we accumulate molecular damage, other negative consequences, we produce mutations, and all kinds of other things. And eventually it would result in functional decline late in life, eventually in diseases, and finally in death.
So the ticks start at about 3 weeks in human equivalent terms.
Based on this clock, aging clocks, yes. It would be, again, so it would correspond to the lowest biological age, and then from that time point, aging would increase.
So would the biological age actually be higher at conception than at 3 weeks?
It seems so, actually. So that's the also one of our findings, actually. Yeah, we found basically the fertilized egg is called a zygote. So the age of the zygote is very low, but it's not zero, slightly above zero. And during early development, early embryonic development, it actually is decreased, especially post-implantation. Like when the embryo implants and in the uterus, then it grows, it begins to differentiate, and then the soma kind of is born. So, and that's kind of marks the beginning of aging, also marks the lowest biological age. It's actually interesting, the way we kind of consider this question is like this. So at the end of 19th century, there was a famous scientist, August Weismann, and he proposed this separation of aging soma and ageless germline. He basically says that the germline should not age, because if it would age, then the next generation would start at a higher biological age, and then the following is even higher and higher, and eventually everybody will be extinct. It does not happen, so therefore germline cannot age. And you can literally find this in thousands of papers, like the statement, germline does not age. And on the one hand, it's true, it should not age. On the other hand, how can it not age if germline is represented by live cells, like oocytes and sperm? And we know, like in older people, for example, sperm is really older in older men compared to the young men, so it does age. So, but this would mean that it also must be rejuvenated. The question is when. So we actually initially thought that the only time when it can be rejuvenated is, post-conception. And so we tested by using biomarkers of aging, and we realized, yes. So initially it's low but detectable, above zero, let's put it this way, and then it's decreased in early life, culminating with the lowest level at gastrulation.
Fascinating. So it starts at a certain age, and then it actually goes backwards or reverses aging to hit the absolute zero of aging, which is during gastrulation, and then the clock starts ticking again.
Yes. This is interesting. It's not just a biological age that we quantify, but again, so I would stress that it's just conceptually, like it must be rejuvenated, so somehow. And we also observe that at the level of, like, for example, telomere extension or DNA methylation remodeling, or removal of byproducts. So it's kind of associated with the preparation of the embryo for like future life. And once it's done, then that's it. Kind of the life begins and aging begins.
And this is all based on some measure of aging, some kind of way to figure out a quantity of age. And how do you measure aging?
Well, we measure aging. Well, it's not easy to measure, definitely. We use so-called clocks, aging clocks, so biomarkers of aging. And this approach was started about 10 years ago by a scientist named Steve Horvath. At the time, he was at UCLA. And he published a paper on the first human multi-tissue epigenetic aging clock. This is the clock is basically a set of cytosines, methylated cytosines in the DNA that he identified out of many thousands. He identified, 300 or so sites. Some of these cytosines increase methylation with age, some decrease methylation with age. And by applying machine learning, he identified this mathematical formula with the value of methylation, the particular cytosine, and each cytosine having a weight in the model. And so this calculation is possible from the analysis of DNA methylation, like genome-wide, and this allows to quantify age. So he trained the first clock for chronological age, but since then, subsequent biomarkers have been trained on phenotypic changes, like changes like in the parameters of the blood, for example, as well as on future mortality. That's also can be trained on. Now this field has grown really dramatically. So clocks are published literally every day. Different clocks trained on different features, multi-species, multi-tissue, universal clocks, single cell, whatever, bulk, all kinds of different biomarkers have been developed.
So could you explain first, what is a cytosine?
Oh, cytosine DNA is composed of 4 nucleotides, and one of them is cytosine. And the thing is that in mammals, in some other organisms as well, but not in all, that cytosine can be methylated. There is a methyl group, so CH3. It can be in kind of 2 states, and some cytosines, they may gain methylation, become more methylated, like in different cells, that particular site, and some would lose methylation with age. And so these changes can be quantified. But of course, biological age also can be quantified based on other features. It's not only epigenetic changes like DNA methylation. We could quantify it based on transcriptomic changes. This is the clock that we also have developed in the lab. There are clocks which are based on the proteome, like proteomic changes in metabolites. So mostly at this point we have kind of single omic clocks, but in the future it's going to be probably multi-omic when all of these modalities are included in a single model.
And when you say transcriptomic clock, what does that mean?
Well, transcriptomic clock is based on the levels of the transcripts. Like there's a genes are expressed, and this is the transcripts or mRNA. It can be quantified by sequencing the samples, like doing sequencing, RNA sequencing, and quantifying the level of gene expression of each of the transcripts. And again, like in the case of DNA methylation, a model can be built, like elastic net, for example, regression, or some other models where we would quantify aging by quantifying the levels of the transcripts, of thousands of transcripts.
So let me just understand this. We have DNA, and then that expresses certain proteins via RNA.
Yes.
And the levels of RNA expression, that's the transcriptome.
Yes, well, I mean, I just don't know what's the level of the audience, of course, but it's like a dogma, major dogma of, molecular biology is we have DNA, RNA, proteins.
Yes.
So ultimately proteins do the work in the cell, but they are made from the mRNAs by the ribosome, and mRNA is synthesized from the DNA.
Yes, and so we're looking at the levels of expression.
Yes.
Because we can have more expression or less expression with the same DNA.
Yeah, in the cell, DNA is present in the nucleus, but then there are genes like 20,000 major genes and various other smaller genes, but then they are expressed at different levels. Some proteins are needed, for example, in larger quantities. Therefore, you would have more transcripts, and therefore more protein would be made from the transcripts.
So the transcriptomic clocks, they work via the intermediary of how much transcription, how much mRNA,
Yeah.
and then the proteomic clocks would be looking at the quantities of the end product.
Of the proteins. Yeah.
Of the protein.
Yes.
Mm-hmm.
But the main feature here is not DNA methylation on the transcripts or the proteins, but it's just out of the many features that are available, it's just more efficient. It's more efficient way to identify features that really represent aging, the aging process. So typically we select those which better represent, and then they become part of the model.
So you have these multiple clocks that use multiple indices. Can you use these clocks to evaluate interventions?
Well, yes and no. If the effect is large, then we can. I remember when we published our first clock, which was a mouse clock, and we trained it based on chronological age of mouse blood. We also, once we developed it, we tested the effect of interventions like a calorie restriction. So in each group of mice that been subjected to calorie restriction, they seem to be younger based on this clock than the chronologically identical control mice. So we could test this intervention already when the mice are, let's say, 10 months old. We already can see the difference. Another example, which we also published in that paper, is the comparison between control mice and mice in which a particular gene, growth hormone receptor, was removed. These knockout mice, they are longer lived. They live maybe 40% longer than control mice. And at the age of 6 months, we already could see the difference. So that growth hormone receptor knockout mice, they are also biologically younger than age-matched controls. So in that sense, the clocks that we developed already could be used to test interventions. But when the effects are smaller, it's not always the case. So at this point, we don't really have like a fully validated clock that would predict interventions, at least in humans. This is still ongoing work in the field to do that.
So which clocks do you have the most faith in, and how do you look at the question of clocks?
I don't have a favorite clock. It just really depends on the question, like what exactly we would like to quantify. If you want, let's say, to predict like an age of a tissue or protein, that we would use the kind of traditional Horvath clock. If we'd like to predict mortality, maybe I would use a clock called cream age. Future mortality. If we would like to predict phenotypic changes, it would be maybe pheno-age. That's another type of clock. There are clocks that quantify the rate of aging. So this is based on the longitudinal analysis when like samples are collected periodically from the same individual, many times, and the clock built on the rate. So the most famous clock of that type is called Dunedin Pace, developed by Dan Belsky from Columbia University. We developed, recently we published a paper on developing the so-called causal clock. This is actually an important also development in the field. And let me actually tell about it. So traditional clocks, whether they are built based on chronological age, trained on chronological age, or future mortality, or phenotypic changes, they incorporate all of the changes that happen with age. So, for example, if aging is accumulation of damage, they would quantify damage. But because the organism responds to damage, activating protective systems, like trying to defend itself, those activated processes, they also show age-related patterns, and therefore they also become a part of the clock. And so when we test interventions, we don't know which part of the clock reports which part of the aging process, and therefore we cannot be sure that interventions that they really change the aging trajectory. So what we really need to do, we need to separate these kind of negative changes that happen with age from adaptive protective changes that result from those negative changes. And that's what this adaptive clock is. We try to separate them, and we found that even though both types of clocks, whether like based on damage or adaptation, they equally predict chronological age, they respond very differently to interventions. So when we try to, for example, reprogram cells to an embryonic state, like we take like adult cells, like fibroblasts, and reprogram them to induce pluripotent stem cells, which are embryonic cells. So the damage-based clock predicts that these cells are young, like embryonic cells. The age is 0, whereas the clock based on adaptive changes does not, which again, would make sense. That would be exactly as we predicted. So in many cases, like when we test interventions, what I would use this kind of causal clocks that we developed. But again, so there are many clocks, and so far I've been only actually mentioning epigenetic clocks. Those are the most famous, most robust. More recently, again, transcriptomic clocks have been generated, proteomic clocks have been generated, though they also gain popularity.
So with the advent of causal clocks, you're talking about the potential to differentiate between something that it's like someone has poor eyesight, so they wear glasses, and generally poor eyesight comes with being older, but the cause of the glasses is the poor eyesight. And so now with the new clocks, you can separate the poor eyesight from the glasses. And you're saying certain interventions remove the glasses, but not necessarily the poor eyesight.
Yeah, that's exactly. So in fact, we can build a very good clock based on the features like you mentioned, like glasses, hearing aid, like number of visits to doctors, use a wheelchair, and so on. Let's say we choose 100 parameters like that. We build a clock that predicts the age of a person, and then we just fire all doctors. There are no doctors, nobody goes to doctor's office, nobody wears glasses, no hearing aid, and based on this clock, everybody is young, right? But they are not. So that kind of example highlights the causality problem. So we really need to quantify aging based on the aging itself, rather than responses to aging, like wearing glasses or some adaptive changes like hearing aid, going to doctor's office. At the molecular level, of course, the same adaptations happen. And so they are part of the clocks that we develop, and we need to split. And this is a very beginning of this research on causality in aging. So again, we just published a paper 2 months ago, the first paper, and we continue to develop even better clocks.
So when you get to the point of differentiating associative clocks with causal clocks, would using those causal clocks open up a way to find interventions that could work in terms of inducing longevity?
Yes.
Or prolonging?
Yes, that's really the purpose of these clocks. Try to identify interventions. Well, not the only purpose. There are many purposes, but an important use of these clocks is to test them in interventions, because currently we actually don't have a single intervention which has been validated to increase human lifespan. So there are interventions in mice. We have maybe 15 to 20 ways to extend lifespan in mice. We can do it in model organisms, like in C. elegans, flies, yeast, and other organisms. But in humans, so far, nothing has been validated. So there are many suggestions that some supplements or lifestyle changes or drugs or diets, they might be useful, and they may, but it needs to be tested. It has not been tested. And of course, it would be unreasonable to do an experiment in humans when we take like 2 cohorts, like control and treatment, and follow them like for dozens of years until people die. It just would be too long and too expensive. So we want to have 2 cohorts and treat them with something, and maybe after a year or 2 or 3, using the validated biomarkers of aging, tell, okay, this particular intervention slows down the aging process. Or maybe some of the interventions might even reduce biological age. So those are the purposes of the use of the clocks. But I would stress that the clocks have not been validated, so there is still ongoing research in the field. And maybe I should mention this, that we also organized a consortium called Biomarker of Aging Consortium that brings together many leading labs in the field, which debate and discuss. We have meetings. There was a meeting like a few months ago that was really well attended, oversubscribed. We couldn't accept everybody who was coming. And the next, we will have a meeting in this November exactly on that topic, on try to help develop biomarkers that could be used by the research community to test interventions and test other aspects of aging research.
But with the advent of certain causal clocks, can aging be reversed if it happens in from conception to gastrulation, can it happen in adult bodies?
Yes. So I already actually mentioned an example. So when we convert fibroblasts, which are somatic cells, to embryonic cells, induced pluripotent stem cells, and this process was discovered by Yamanaka, and that's for this reprogramming we use so-called Yamanaka factors for transcription factors. And what Yamanaka found is that the cells can be converted from like adult state to an embryonic state, but for us this means that the cells were converted from an older state to a younger state. To me, this is the best example of rejuvenation, when we start with older cells and end with young cells upon expression of 4 Yamanaka factors.
Do you know of other ways to rejuvenate cells or reduce age?
Yes, so we also published last year a paper in collaboration with Jim White at Duke, and this paper dealt with the method called heterochronic parabiosis. So this is a kind of complicated name, but basically what this means is that we take mice, like one old mouse, one young mouse, and we connect them so that they have a joint circulation. They exchange the blood. And the young mouse, we found, actually rejuvenates the old mouse. So we keep them connected for 3 months. After 3 months, we separate them. And 2 months later, we test whether the old mouse is still rejuvenated, and it is rejuvenated based on biomarkers of aging that we discussed. But when we looked at the lifespan, they also turned out to be longer-lived mice. So this is a very nice and robust example of rejuvenation of the organism. The effect is not huge. Of course, we would not be able to rejuvenate all aspects of aging, but the biological age seems to be reduced by this process, and also it makes them longer-lived.
And the young mouse that was discarded as lived shorter?
So with the young mouse, we published a separate paper on this topic, and we found that the young mouse rapidly becomes older when it's connected to the old mouse, and when we separate the mice, they recover. They're able to remove the damage that accumulates in the mouse, and kind of the age is restored. So yeah, this is a very interesting model system, actually, that can be used to study various aspects of aging.
So the young mouse bounces back?
Yes, so we cannot be sure it bounces exactly to the control level, because the accuracy of the clocks is just insufficient. All we can say just rapidly increased age and then reduces approximately to the expected level, but we cannot be sure it's exactly, like, maybe slightly above. I don't know.
Well, if we don't want to tie our circulation to a younger person, or vice versa, yeah, are there any other approaches to rejuvenation?
So yes, we also discussed, like earlier already, this when we discussed the beginning of aging. So this phase from the zygote, which is a fertilized egg, to the gastrulation, this lowest point of biological age, we call it ground zero. So this period from the zygote to ground zero is actually a phase of rejuvenation. And we discovered it recently and reported in several papers. So we don't understand mechanistically how exactly this happens, but we observed this in mice, in humans. We observed in some cell culture models, and we have some preliminary data in frogs as well. But this is an ongoing area of research. It needs to be validated by other labs. But it seems to be the first natural process of rejuvenation that happens during early embryogenesis.
And how do you see any of these approaches being translated into therapies?
Well, what we discussed so far, clocks, biomarkers of aging, they can be used mostly to test interventions. But to predict interventions, we need something else. We also work on this in the lab, and for this we developed so-called longevity signatures. So what is longevity signature? It's basically an omic patterns that represent the potential of the system to live long. For simplicity, let's compare like human and mouse. Mouse is a short-lived, human is a longer-lived. How can we modify the mouse so that it is lived as long as human, or at least longer-lived? So of course, if we only compare human and mouse, we will not be able to do it because we immediately will identify human-specific and mouse-specific genes. But instead, what we do, we compare many species. Like the paper we published last year in Cell, we compared 41 species of mammals. So we analyzed them at the level of gene expression across multiple representatives and multiple tissues of each species. There was a very large kind of study. And we identified in every organ, we identified several hundred genes that represent this potential to live long. Yeah, basically, we ask a question, how does nature extend lifespan? Across mammals, there's a huge diversity in lifespan, over 100-fold. Some shrews live maybe 1 or 2 years, and some whales can live up to 200 years. So we are all mammals, but somehow nature is able to change something to achieve this huge effect, radical changes in lifespan. Instead, in the lab, what we study, the effects of calorie restriction or rapamycin, other interventions, are relatively small, 20%, maybe 30%. Across mammals, we're talking about 100-fold, right? And so these tools that we develop, whether it's cross mammals or across interventions, we call longevity signatures. Again, so they're indicative of the potential of the system to live long. A third component that we use is comparison across cell types, because cells within the organism, they also have widely different lifespans. Some cells, like neurons, they live the whole life. They are born embryonically during embryogenesis and live the whole life. And some cells, like cells in the blood, they are changed every few days. They are very short-lived. So by analyzing many cell types, we also can define principles of lifespan control across different cell types. And then we integrate all of these signatures across cell types, across interventions, and across mammals to basically develop actionable markers of longevity. We call them longevity signatures. And then we apply these signatures to identify interventions. We can screen databases, for example, of drugs, and ask a question whether any of the drugs, they mimic those longevity signatures. I would say responses to these drugs would mimic longevity signatures. And now we have a large project in the lab where we identify these interventions and then test them in the mouse model. And so far the results are very encouraging, but this is still unpublished work, so maybe we'll discuss later in a few years.
So you've been looking at the natural ways of increasing longevity, or the natural dispersion of longevity, and then working backwards and seeing how different interventions could mimic that, and then seeing if that intervention actually does mimic it in a whole organism.
Yes. But because we have different omics patterns, different signatures, we also found, actually, there are very interesting observations. So for example, if we look for signatures that mimic known interventions, like calorie restriction, rapamycin, canagliflozin, acarbose, or some generic models like growth hormone receptor knockout, and so on, we can easily find new interventions. In fact, many of the interventions that we observe in the lab, they already are similar. For example, methionine restriction or every other day feeding, they're similar. But when we look across mammals, it seems like not so many interventions. I would say almost none of the interventions mimic that signature across mammals. So it's actually a very interesting realization that whatever we do in the lab is not the same as the interventions that we find in the lab are not the same as what nature finds when it extends lifespan. And so this represents a really untapped potential for radical changes in lifespan if we can find some interventions that mimic what nature does. Of course, it's very difficult to do because natural changes in lifespan involve many genes. It's never a simple process like one gene knockout or one intervention. It's a very complex combination of factors that nature uses. Whether it may be possible to recapitulate this experimentally or not remains to be seen.
Is there a way to boil it down? For example, when you see a cluster of signatures that seem to indicate an expansion of longevity, and maybe that involves, I don't know, 1,000 genes, is out of those 1,000, can you find the causal ones, the most important ones, the ones that carry the weight?
Well, this is also something that we're currently working on. So we don't have answers yet to this question, but hopefully in the future we will have. The causality is really key in this kind of studies.
And these causal clocks, they all start at the minimum age, not of the cell, but of the organism, yes?
Yeah, so that's another issue actually. Like, how do we study aging or quantify aging? So far we mostly quantify at the level of organisms. But of course, organisms are composed of various organs and tissues, and organs are composed of different cells. And organs and different cells may age with slightly different rates. A couple of years ago, we actually published the first aging clock that quantifies age of individual cells, the clock called SCH, for single cell aging clocks. And so by applying this clock, we can see a huge heterogeneity in the ages of different cells in the tissues. Not huge, but there's some heterogeneity. We also found that some stem cells, they age slower. Not all stem cells, but some, they age slower. And some cells, which are maybe more damaged cells, they age more rapidly within the tissues. Also, there's a recent development actually in the field on quantifying aging of tissues. And one really excellent study was published a couple of months ago by the lab of Tony Viscoray from Stanford, where they used proteomic clocks to predict age of different tissues based on the patterns, proteins in the plasma in humans. So we also have actually a manuscript on that topic where we've also developed proteomic clocks that predict the age of different tissues slightly differently. We use a different data set, and also we train the clocks based on mortality, future mortality, rather than chronological age. But I think this is a very, very interesting development in the field. So why is it exciting? Because what we think is different organisms, when they age, some of their organs might age more rapidly. Let's say all of the organs age with expected rate, but let's say the liver ages faster. So this person is more likely eventually to develop liver disease, some kind of liver cirrhosis or maybe cancer. And therefore, this would be like a weak link that may potentially be targeted. So this kind of idea of organ-specific anti-aging interventions is a really novel idea, and it actually has not been done yet. And we are looking forward to these advances in the future.
That's fascinating. So if you have a heterogeneity of ages in your body, so an organism will have a 10-year-old cell according to its biological age and a 40-year-old cell in the same organism, theoretically. When does the organismal life begin? When does the organism start aging as a whole?
I think the way I see your question is could be addressed from two different angles. Yeah. So one is the diversity of aging patterns of different cells within the organism. So one interesting observation we have is that cells are not in isolation. Cells and organs, they talk to each other. They interact, and so they influence aging trajectories of each other. So let's say if you have a old organism, and then there's a young organ, let's say, in that organism. So this organ would rapidly age by the old organism, but it also would influence other tissues in the body. So, and these interactions become very complex. So sometimes not easy to disentangle these interactions. Your kind of second angle of your question is you kind of mentioned on the beginning of life, and that's a whole different kind of perspective, which is maybe controversial even to some degree, because it's actually an interesting question. So when does life begin? Organismal life, I mean, the cellular life begins at the stage of conception. But to me, organismal life begins later. So why I think so? Let's say early in life, you have a 2-cell embryo, or 4-cell embryo, 16-cell embryo, called a blastocyst later. So at those stages, you could separate cells. Like you have 8 cells. You remove one cell, and that cell will become an embryo. That's how twins are formed, for example, naturally. These are 2 lives. If like one life began and then split into 2 lives, that would be strange. And also at that point, we could not just combine embryos of different embryos, like we could basically fuse embryos, it would become one organism.
Mm-hmm.
We could even fuse cells from different species. Like, for example, we can take mouse blastocyst and inject rat iPS cells, induced pluripotent stem cells. We would have a so-called chimeric animal composed of both mouse cells and rat cells. And this is also a very exciting kind of direction in the field when chimeric animals can be studied composed of different tissues. Perhaps you might heard of the ideas of generating like human organs in pigs, like for example growing a human heart. That's exactly is done through the chimeric strategies. But in terms of life, I think that early in development, this phase really represents preparation for organismal life. And then when the damage is cleared, when like somatic soma is born and at least separated from the germline, when individuality of the organism kind of develops, when cells differentiate to 3 germ layers and organogenesis begin, that's, I think, the beginning of aging, but in a way also the beginning of organismal life. At that point, you know, we can no longer combine like cells from different species or like 2 different embryos of the same species. There is already the level of individuality that happens. So, but again, so this is at this point is pure speculation. Is the way of thinking, the way we think about these processes. But scientifically, it's a little bit hard to study because we need to define what is life, what is the beginning of life, and those questions are not well defined.
So the beginning of organismal life, in your opinion, is when there's the start of organogenesis, and that's the start of aging as an organism, whereas the clocks would indicate that the ground zero of aging is at the moment of gastrulation, which is in humans about 3 weeks after conception.
Yes. So it's all of this because it just happens approximately at the same time. The beginning of aging based on the lowest biological age and beginning of accumulating damage. It's the birth of the separation of the germline and soma happens at that point, and the differentiation into germ layers and the beginning of organogenesis all happens during the stage of gastrulation. That's, I think, is a very critical stage. There was a scientist, I forgot the name now, but there's a famous phrase that he said, it's not, I can't remember exactly, but it's not your marriage or birth or death which are most important phases in your life, but it's gastrulation, which is the most important, which I fully agree.
So given your depth of knowledge in clocks and aging, in the potential for rejuvenation and decreasing biological age, what do you personally do to decrease your biological age or rejuvenate yourself?
Yeah, I wish I would know what to do. I literally do nothing. So because I don't know what to do, we know how to shorten life. Like, smoking is really bad. It shortens by 10 years. Drinking, alcohol consumption in large quantities is really bad. Sleeping insufficient time is really bad. Excessive stress is really bad. But if you have a relatively healthy lifestyle, if you eat well and sleep well and don't drink, don't smoke, and I try to follow this, then what else can you do? That's a question. I don't know. I would stress that currently we don't have a single intervention known for sure to extend lifespan. So I think it will be tested and we will find some. There are many good candidates, but before they are validated, we cannot really recommend any of them. So maybe in a few years we will know, and then I will start following this, but not yet.
Well, hopefully we'll have them soon, and you'll come back to tell us about it.
I will.
Thank you so much for joining us.
Okay, thank you very much. It was great, interesting discussion.
Thank you for joining us today. We hope you join us next time as we examine and elucidate the frontiers of science and technology with the thinkers creating our tomorrow. Until next time, farewell.